Okay, hello everybody, and welcome to the first seminar for the Ecology and Evolution seminar at the University of Oregon from fall of 2020. So today's seminar will go as is the usual plan for this quarter, which is remotely from 12:15 starting at 12:15 for the usual 50 or so minutes with questions, then a short break, and then a half an hour sort of coffee chat with the speaker. And everybody's welcome to join that. If you're watching on the live YouTube link, then just email me, Peter Ralph, for plr@uoregon.edu for the link. Okay, and right now I've got everybody muted. I will probably make it so you can unmute yourself, but keep it muted unless you're asking a question, or if that doesn't work, then go ahead and say something in chat. I'll try to keep an eye on that. Okay, so we are pleased to have Yaniv Brandvain from the University of Minnesota speaking today. He's in the Department of Plant and Microbial Biology. He got there via the College of Atlantic and the University of Indiana, where he worked with Leonie Moyle. And I think it's fair to say that Yaniv is one of the foremost thinkers in population genetics today. He works on a variety of things, doing really great data analysis, evolutionary theory, teaches a mean statistics class. Yeah, but I think it's fair to say that the things that get him most excited are plant mating systems and genetic conflict. Is that fair to say, Yaniv?
Yeah, I get excited about everything. I think one thing I really like is that plant mating system, hopefully I've convinced you, allows us to see so much of all these questions that we all care about. So I find it is a great way to jump into these big questions.
And on that topic, I think that's what he's going to talk about today. So take it away, Yaniv.
Okay, thanks, Peter, and thanks everyone for coming. This is my first virtual visit, and I was just amazed by how fun it was and how I got that same sort of excitement and charge and joy that you get out of talking with people at a standard visit. And so that was awesome, and I really appreciate all of your time. I know we have a lot going on. And I'm here to talk to you, as Peter said, about lots of how I consider myself somebody who thinks about a lot of things. And I often think that it's easy to think about, who cares how plants mate? It's these weird things, and you boring, I don't know. But I really think that this is one of the most exciting avenues to learn about all these big questions in evolution. And so I think that, and in ecology, so one of my main goals for whenever I give a seminar is to try to convince you that this stuff is super cool, kind of no matter what you care about. So hopefully there'll be something in it for everybody. And these have been, as Peter said, I use a lot of different ways to answer these questions. Okay, so I think that by studying, how plants reproduce, we can get at some of these big questions in evolution. What's nature of genetic variation? How do various evolutionary processes shape genome structure? Why do organisms live where they do? Where do new species come from? What are the major, all these things that we care about, I think we can get at by looking at how plants mate and their consequences. So that's what I'm going to try to talk about today. Okay. And as Peter said, I do that through whatever data sets I can get, right? Or whatever data sets are appropriate to the question at hand. Sometimes that's simulated data or some theoretical results, theoretical data. Sometimes it's meta-analyses, comparative studies, whatever we can do to answer these questions, population genomic studies. And, we put this all together just to try to think about the world. Okay, and so as I said, the big goal is to have you think, man, this is cool. I have some question about biology. Maybe I can use how plants mate as an opportunity to address these questions. I just want to show you that it exists. There's lots of variation out there. and then show you a few different consequences about, what this does for plants, how this might impact hybridization, introgression, and then think about some tensions that plants might have when they mate. We might be familiar with all of these things. And so the first thing I just want to discuss is that there's just a tremendous amount of variation in how plants mate. And this allows for tests of theory. And when we think about how plants mate, we usually think of it as sort of two extremes in terms of plant mating systems. On one extreme, you're mating in a somewhat large panmictic population with whoever you bump into, right? And that might be one of these large scale dating apps. And then in the other extreme, maybe you just happen to be mate with whoever's around. Often that's close kin or inbreeding. And that's sort of the two extremes by which we often think about the plant's mating system. And in plants, it's even more extreme than mating with siblings. Often it's mating with yourself because plants, don't separate their male and female parts. Okay, and so what we see is that self-fertilization seems to be actively discouraged in many plant species. So they go through elaborate processes. Either they have their their parts sticking in different directions, or they separate the time at which they release their pollen from when their styles are receptive, or they have chemical systems to think like to prevent self-fertilization, which we'll talk about shortly. But yet other species, often in close relatives, it seems like plants go out of their way to mate with themselves. So you'll have two close related species. In one case, they've gone through all these elaborate mechanisms to avoid mating with themselves. In other cases, they don't even ever open up their flowers, right? Or their pollen, their flower development is such that they just lay their anthers right in their stigmas. And this histogram that I'm showing here is somewhat controversial. People have argued about it, but it seems like, you can see that there's plenty of plants at each extreme here in terms of plants that are fully outcrossing or largely outcrossing, and plants that are largely selfing. And then at the other end, both when we think about, at the most extreme of mating dissimilarity, you can even have some plants that mate between species. And we'll talk about that later. And that's also somewhat common in plants, and we're learning more and more that we see that in a bunch of organisms. So it used to be, I don't know, back in the other 20s, people would think, oh man, plants are so weird with their ability to, mate so widely with so many different species. And now that seems to be becoming more and more the rule across a lot of taxa. Okay. So what are the consequences? One thing we'll talk about is self-fertilization leads to less conflict between genomes, right? If you've inherited genomes together, there's less for them to fight about. Self-fertilization makes genetic combinations heritable. And so you can select on all sorts of components of genetic variation that you usually can't see in panmictic populations. You can selection X differently because of differences in the effective population size and differences in whether recessive mutations are exposed when rare or not. And there's all sorts of other ideas about what might happen here. And so I'm going to start with an example, and this is an example of the young me. So this was one of my first science projects I did as an undergrad. I thought about this. I really got excited about this idea about tensions in the genome, fights between mom and dad, and I realized that selfing and outcrossing species have the opportunity to shed some light on this, right? Because when you're mating with oneself, there's much less opportunity for fights over resource allocation to the kids, right? What's in the mom's best interest is in the dad's best interest if the mom and the dad are the same plants. By contrast, if the moms and the dads are different plants, there's room for conflict, right? And there might be some disagreement over resource allocation, where with outcrossers or with the mom saying, I got more kids to feed, and the dad, saying I need to do all right. And you see this often played out in development in these genes called imprinted genes, which are differentially expressed based on which parent you get them from. The overall pattern is just as you would predict. It seems like genes expressed when inherited from moms tend to be growth suppressors, and those inherited from dads tend to be growth enhancers. And one of my first science projects, I read all these old scientific papers and said it seems like you can see evidence for the sort of footprint of this conflict when you look at crosses between self-compatible and selfing plants and outcrossing plants. And so when the mom is from a population that's traditionally outcrossing, and you cross that to a dad that's for a population that's traditionally selfing, it seems like the mom is winning. She's basically pulling harder in the tug of war, and so the seeds are smaller. And that's usually manifested through endosperm development, and the endosperm is a little smaller. And the reciprocal cross, the dad seems to be winning in that when the dad comes from an outcrossing population, those seeds, the endosperm gets so big. It's not actually a win because they're so divergent. Often these plants have trouble developmentally either way, but it's a way to look into this conflict. And so this was just to say, this is an idea that got me started, and it made me think about the different ways we can leverage data out there and crosses between selfing and outcrossing plants um, to try to test these broader evolutionary ideas. And one thing that's been interesting for me as I've developed as a scientist is that there's more and more convincing data from from transcriptome studies and so on that seem to be looking at both replicating this pattern and getting a better mechanistic understanding of it. So now moving to some of my dissertation work, just say these ideas have been with me for a long time, is that it's really cool, right? If you take a quant gen class, if you ever read Fisher and think about average success versus average effect, right? You can know that in general, natural selection only gets to see the additive component of genetic variation. But if selection doesn't randomize those, then you actually get to see sort of the marginal effect. And so because selfing keeps gene combinations together, you can more effectively select on epistatic variants, which you can't do when you're outcrossing. And so we use this idea to develop some theory about how you can move genes between the nucleus and the mitochondria, which is a coordinated, genetic system, and thought you should be able to get more gene transfer when you're more selfing, and data from literature appeared to support that. Okay, so now moving to the more recent present, selfing exposes inbreeding depression and purges it, or may or may not purge it. And so this is some fun work that just came out yesterday, so I didn't update all these from bioRxiv to Evolution Letters, where Kathy's work just came out. But Kathy was looking at different, who's a postdoc with me, is looking at different Bucra species. One that's selfing, and then there's lineages that are kind of permanent heterozygotes. And she found a bunch of evidence that that the selfing lineage had a greater had a tougher time living, presumably because of inbreeding depression. But she also found that the permanent heterozygous combinations had their own issues. Okay, and then the final result I want to talk about is something that I'm just scratching my head with. And this is something that I can't say I understand. This is the me from, went from me from an undergrad to me as a grad student to me yesterday. Or in me yesterday is basically the brains of my fantastic postdoc, Shelley, who was looking at this classic question about how selfing should impact the ability of plants to get rid of bad mutations or accumulate them. The classic idea is that because selfing species are immediately identical by descent at all chromosomes, right? Because more or less, because you just, self, you're all the same. Your effective number of chromosomes of that population should decrease by a half. And therefore, selection should be less effective at removing mildly deleterious additive mutations. I've seen this thing cited a million times. Shelley wants to do a fancy and exciting simulation project in SLIM that I would love to talk to you about later, but we just couldn't get this result. So she just couldn't find this. So here's some of her results. We have, this is actually, we just went back to the basics and did a very simple forward simulation. We introduced the mutation of frequency 1/2n and looked at its probability of fixation. And the line of 1 is where I scaled where it should be if it were acting as if it were nearly neutral. Here's different selective coefficients. So at the top left, this is something that should be acting as if it's nearly neutral given our population size. At the bottom right, that's something that should be pretty close to being, uniformly rejected given our effective population size. And the x-axis is the selfing rate, and you can see that there's almost no impact of the selfing rate on the fixation probability. You can see there's subtle effects with the dominance. So if I'm totally recessive, selfers seem to fix a fewer mildly deleterious mutations. If I'm additive, there's basically no pattern, and if I'm um, dominant, there's a slight pattern in the opposite direction. And this still looks almost nothing like the twofold expectations that you see directly under there. Okay, and so I'm just pointing this out, first of all, because I want more eyes on this result, because I'm so confused. I've read a million papers that have said this. I can't find evidence for this. So if anyone can figure it out, please tell me. Why else am I saying this? It's weird. And I guess as we get more and more data and as we get more and more sophisticated population genetic simulations, I think we're just learning more and more that the fundamental theory that we've done with math doesn't always work, that this arbitrary term called the effective population size in one equation doesn't always plug into the arbitrary thing called effective population size in another equation. And this is just an exciting lesson learned. So uh, just sharing that with you. And one more thing to share about with Jelly's stuff is that there's this other idea that selfing can decrease the efficacy of selection because there's less effective recombination. You keep genes around, you kind of reduce the effective population size longer across the chromosome as you remove bad mutations. And we find that that only happens sometimes and is particular to your parameter choice. And there's lots of ways to get that result and lots of ways to get the exact opposite result. And so the thing that's exciting for this these results to me is that there's this fundamental theory that I've started 12 papers with. Every time I write a paper, you write these words in the beginning, and we're finding that they just aren't right. Okay, so with that, I first just want to say thank you for bearing with me. That's well, you're gonna be bearing with me all day, but that's sort of the fire hose of here's a bunch of exciting things that have got me started, or a bunch of different ideas about why mating system is cool, about how we can use it to interrogate some of our fundamental thoughts about evolution, like ME, or genetic conflict, or what is and isn't selectable and heritable. And so now I'm going to talk a little bit about how you mate can impact where you live. And so if you're interested more in ecology and species distributions, you can say, okay, I'm glad we got that NE stuff out of the way for a minute, and you can listen to this. And yeah, again, I wish you could interrupt me and tell me things, because it's sometimes hard. Peter, give me thumbs up, thumbs down, okay? Okay.
Great, yes,
Peter.
I can hear you.
Okay.
I even turned on my video, so maybe you can have a look at a face.
Yeah, I see you. Okay. So there's different ways that selfing can influence the species range, and we're going to talk about these. And these were ideas I just started. Lots of them were when I started my job, and you'll see that lots of them were led by a fantastic postdoc in my group, Dina. And so one idea that's very simple is that islands could be enriched for self-compatible species simply because they can live there when they land there. That might have consequences for what we see on non-islands, like on continents. And you might also think about if selfing allows species to live in the same place, because they can then both exist without interfering with each other's reproductive abilities. Okay, so these are 3 empirical projects that made up much of Dina's postdoc with me, and then she is now a faculty member at Cal Poly San Luis Obispo, and it was super fun to have her around leading this work. Okay, so I'm going to cut to the chase and say that islands are enriched for self-compatible species. This is theory you don't need any math for, or not much, right? If you have a plant land on an island and it can self-fertilize, oh, I lost the slide. It can make seeds, right? And if it can't, and it's all alone, it can't. You can do some theory to get more fancy predictions, but it's basically that simple. And so, this is so obvious, right? The logic is so obvious, the pattern is so wide that we should just call it Baker's Law. But this had a few anecdotes and no stats. And in fact, there were a few people that argued vociferously that data wasn't supported by more evidence, or that claim wasn't supported by evidence. And there were no real strong tests. There were case studies, this example and that. Okay. So Carl Quest, who's another botanist, said this must be abandoned. Okay. Before I get too deep into this, I straight this here because I have to take a step back and say, why do we care, right? If you're a plant biologist, maybe you've spent time thinking about Baker's Law and this is really cool. But if not, you might say, okay, this seems like an obvious prediction. Why would anyone care? Right? And the reason I think we care is we want to know, for example, how plants or how organisms in general colonize. Do they do that as single individuals? Do they do that as rafts of individuals, related individuals, or not? And so that might fundamentally have to do with how invasion works, how new organisms get established, or whatever. And so I think that's some of the logic that drives us to think about these questions beyond that sort of simple history. Okay. So this work was led by Dina and some fantastic colleagues who did a lot of work. This is actually an entire part of a working group, but if you've been to working groups, you know that there's certain people that are the true heroes, and here they are. This sort of work requires extensive QC and care that's hard to communicate until you do it when you just see a plot. So I just want to give a few seconds to acknowledge the hard work that these people did as they led the project, validating these records and all that. And so what we see is support for Baker's Law throughout. And so these little pie charts are the frequency of self-compatible or self-incompatible organisms or plant species. And you can see from this, I forget what they call these plots, but that there's a relative excess of self-compatible plants on islands in the Asteraceae relative to the mainland. And you can see a similar but more subtle effect in the Brassicaceae. All of these are quite significant, right? And a similar but subtle, again, subtle effect in the Solanaceae. And so what we see is that there's really strong support for Baker's Law. We see it over and over again. In these meta-analyses, we find that there is indeed an excess of self-compatible plants on islands, but this is just statistical, right? So it's not like that's the only way to get to an island. It's not like a self-incompatible plant has no way of making it. And so this might suggest it's time to think about more serious theory, like how long organisms live, what's the right, how do organisms invade islands, and so on. Then we might be able to fit these, extract more from these data. So we definitely see this pattern, but there's definitely more excitement going on that one could dig into. Okay, so we see that how plants may impacts whether or not they're found on islands. That's cool, but so much of the world is not an island. Or, we live on these vast continents. And so we want to say then, how does this impact the species range? Okay. And so it could be that selfing species have larger ranges because they can, move at the edge. They don't have at the range edge. They don't have to wait for new relatives to show up and fertilize them. When they colonize, they can keep doing so even when they show up from one seed. But on the other hand, selfers are known to sometimes be ecological specialists, be less competitive than their outcrossing relatives. And the data, at least before we got more into it, seems somewhat mixed. And so this was a real hypothesis that was out there. Again, it's a similar crew of people. This wasn't through the working group, but again, Dina led us, Ryan, who's a postdoc here with my colleague Dave Moeller, and Emma Goldberg, who was a professor here with me. We did this together. And so what did we find? We found that selfers do indeed have larger ranges in their outcrossing relatives. And we found this to be pretty consistent. So basically almost every genus we looked at, we found that. We also found that in selfing species, the range seemed to increase with divergence time from their closest relatives. Well, we found no such pattern in outcrossers. And finally, we saw that selfers occur at higher latitudes. And so our interpretation of this is since the last glacial maximum, sulfurs have been able to expand more rapidly across the continent than outcrossers. Okay, and so what's that mean? Right, it means not only that sulfurs have this colonization advantage in this island environment, but that this might translate into their ability to colonize and live in continuous space, or at least in a world that's more like much of what we occupy. Okay, thanks everybody. Let me get some water. Deep breath. Okay, there we go. So the final biogeographic question we're going to ask is: can patterns of mating impact species distribution, and particularly can selfing allow for range overlap? Again, the familiar crew of heroes here. The basic idea is because selfers and outcrossers won't really compete for pollinators. They're rarely will they pollinate one another, and so they should be able to co-occur more often than like two outcrossers. You might even expect the two selfers could co-occur more often as well, because they similarly won't be getting in each other's way. And this was inspired somewhat by Dina's fantastic dissertation chapter before she joined me, where she found strong evidence for this in one mimulus subspecies, or mimulus species pair. So we got data for 98 sister pairs from 20 genre and 15 families. And long story short, we found no evidence, no evidence for this pattern. So that's kind of cool. 2 out of 3 isn't bad, right? What's this mean? It means that species might figure out ways to live together when they're in the same place or not, and that presumably their reproductive interactions only make up a small portion, a small contribution to whether or not they can coexist. That maybe that does something on the margins. Maybe Dina and a few other cases that we saw anecdotally are true and real, but that you would need a lot of power to see it. And more often, there might be vegetative competition or all other sorts of ways that, organisms can exclude one another or not. Okay. So we've talked about how selfing impacts the genome. We've talked about, or outcrossing. We've talked about how organisms might interact when they come into contact. Now we're talking a little bit about hybridization, introgression, and the mating system. Okay, so hopefully you're with me. We've learned from genome studies that gene flow between close relatives is quite common. And so what can we learn when we look at hybridization between selfers and outcrossers? And how can we relate this to some of these bigger questions that I think we're all interested in now, as we see that introgression is more common, which is what are we selecting against when we're selecting against hybrids? It appears in general that gene flow is unidirectional from selfers to outcrossers. So I found this in Mimulus nasutus, the sulfur. I found bits of its genome in the background of Mimulus cutatus, its outcrossing relative. You can see that from these dips and these bimodal peaks of divergence between the species, and from these haplotype chunks that you can find here of the sulfur in the outcrosser background. This is slightly more subtle, but we also find this in Capsella grandiflora. This is work with Tyler Kent and a former undergrad with me, Christoph Stankiewicz, where we find that these blue samples are more grandiflora samples that are sympatric with rubella, are slightly more closely related to rubella than are these lighter blue ones that aren't sympatric. And we can get about a 4.2% admixture portion genome wide, which is roughly what we find when we like tag these haplotypes and do stuff. So it seems like there's substantial admixture. If you look at the literature, we often find similarly gene flow from selfing to outcrossing species, and we're going to ignore that. So that's what we generally seem to find. So what does this mean, right? What happens? What are we selecting against when we select against incompatibilities? Against gene flow, right? One could be you're selecting against incompatibilities, right? Another option is that you're selecting against specifically locally adaptive mutations that are good in one environment and not the other. Or alternatively, it could just be that selfers or outcrossers are just worse because selection is less efficient. I'm not going to answer those questions today, but these are questions that I think the whole field should wrestle with, right? I think lots of us are spending lots of time trying to figure out how we can tease these hypotheses apart. We find slight evidence for selection on incompatibilities in Capsella. So these two chromosomes, 2L and 7L, have these two arms have different bits of an incompatibility, and the bit that makes you compatible from rubella seems to be have more of that chromosome introgressed, and the bit that seems to be more incompatible from graniflora seems to have less. So that seems to make sense. In our studies of Nasutus and Gutatus, we seem to find more introgression with more recombination, suggesting that most of the genome is near something that shouldn't introgress. And if you can get away from it, then you can introgress. And we find similar patterns with both recombination and gene density in Capsella. So in Capsella, where there's more gene density of less introgression, right? Because maybe you're near something that matters, and with less recombination, you find less introgression. So we don't know what drives these patterns. This is a question that we don't quite have our hands on, and I think the whole field wants to address it. Okay. But we do know that often selection or gene flow is disfavored, and that there can be some tensions over that. So I'm going to tell a little story. We have time, yes, about how that plays out in interactions between the pollen and the pistil. So this is a project that's inspired by real world data that had us scratching our heads for a while, of what we see when we have maize and its wild relative teosinte, different subspecies of the species Zea maize in the field together. So these species are distributed across Mexico, and there's a highland variety of maize of teosinte called Teosinte mexicana, and it comes into contact with what they call local land races that local farmers are growing, and there's all sorts of excellent work, much of it by my collaborator in this project, Jeff Rossibara, who's looked into some of this. But one thing that's super cool about the system is that despite that there is some gene flow, there seems to be this system of pollen style and compatibility available for them to use. And so how the system works is that there's two tightly linked loci. We'll call one M, and that's expressed in pollen. And if you have that big M expressed, you can pollinate anyone. You have a complementary thing in the pistil, we'll call that F, big F. If that big F is expressed, you require that big M for fertilization. Otherwise, anyone can fertilize you. And so the only cross that doesn't work here is when you try to put little M pollen on a big F pistil. Okay, so that's the biology. There are somewhat surprising patterns of geographic variation at these things. And our sort of interpretation of this, if you kind of take a step back and look at this map, you don't have to, but is that it seems like these big F alleles, these alleles that reject foreign pollen or reject little M pollen, are quite common in teosinte when they're found in sympatry with maize, suggesting that teosinte evolves to reject maize pollen. But whenever that happens, it seems like maize has the big M allele, which allows it to overcome that barrier, and it seems moot. And so this confused us and led us to develop some theory to try to figure out, so why is teosinte evolving this ability to reject pollen that doesn't seem to exist? Okay, and so we developed a model inspired by this. So we had a styler barrier, right, that we started to be rare in teosinte, and we said it would reject maize pollen. We have this pollen allele, this big M from Mexicana that we seem to see throughout Mexicana's range. And so we started with that being quite common in Mexicana. but we don't see that in maize when they're allopatric. So we started that not existing. Okay, and so we started with a maize population that had no style barrier and no style incompatibility or pollen compatibility. While in teosinte, they had the styler incompatibility was quite rare, and every teosinte plant could pollinate that, could overcome that barrier. We also then put down a locally adaptive locus, something to keep these two species apart. And they were in two different populations and they were exchanging genes. Okay, and so we just did, what evolution does. We had pollen that moved between populations. They had kids, they lived or died according to their fitness. They recombined across this thing, and we just watched what happened. Okay, and so this is a small description of our model where, here's the compatibility rules. You again start with these different genotypes in teosinte and maize. So maize starts to be all without the pollen or the styler allele, while teosinte begins with having all males compatible and a few styles are compatible. We let that evolve, and we found two qualitative outcomes. Okay, what we tended to see is that initially we would see something like the F allele, the incompatibility allele, rise to really high frequency in teosinte, presumably because it allows it to reject foreign maize pollen. They can't pollinate it. But then with time, the big M allele, the compatibility allele, spread through maize because better for maize to pollinate and sire low fitness hybrids than to totally miss out on that fertilization opportunity. So that was our rough qualitative result. And then we can think about, more quantitatively how strong does selection need to be? When do we see these different outcomes? What allows introgression, what allows this process of reinforcement to last longer or less long? And so basically when selection is really strong, this can happen more easily and last longer. But really strong, these are values that are just massive, right? This is just one locus selection of 0.5. This is just really, really kind of, I would say, well, maybe it's realistic because who knows, these are divergent species, but pretty intense. Migration didn't have a directional effect, so it seemed like intermediate migration overall was most permissive to allowing this reinforcement to happen for some amount of time. We found that in general, selection in maize and the strength of selection of maize did much more to modulate how much reinforcement could happen and for how long than selection in teosinte. Here, the idea is, right, it's always good for teosinte to reject maize pollen because it leads to bad hybrids, but the relative benefit of accepting of overcoming that barrier to maize is moderated by how bad their kids will be. And so if their kids aren't so bad, they do better to overcome that barrier. So we allowed for asymmetric selection there. Similarly, with asymmetric migration, results were much more sensitive to the rate of gene flow from teosinte to maize. Again, this is a way to think about how much maize gets out of this. If those maize genes are stuck in the teosinte environment forever, right, they don't get much benefit to overcome that barrier, but if they have a chance of getting back to maize, that does better for them. Okay, and the final result that we found is that you need tight linkage, particularly between the locally adaptive allele and the male pollen compatibility allele. So this is super cool, but we got a little bit depressed because we need really tight linkage between the locally adapted allele and the male pollen allele, and because you need selection to be super strong. So could this explain anything? I don't know. If you look, there's 3 such pairs of loci, these they call gametophytic factors, across the maize genome. And you can look at one and say, oh, it's by some allele that might be locally adaptive, but nothing that super bites you in the face or whatever. But what we found is that actually with more unlinked loci, you can actually get a similar pattern, even if there's no physical linkage between the locally adaptive loci and the pollen barriers or the reproductive barriers, simply because then you start to get caught up in the pedigree. And so that was pretty cool. We've only done this with a handful, like 5 loci. We haven't gone fully polygenic, but you can see that you can get now pretty strong results with still quite strong selection, but not the same levels that we needed before. All right, so the really cool implications, right, is that reinforcement should of these post-mating prezygotic barriers, they should be transient, right? That's a new prediction we hadn't thought about before. Or maybe you have to invoke some trade-off that means that it costs May something to overcome this teosinte boundary. And so that there's some opportunity cost that is often overlooked in some of these models of reinforcement, or sometimes they're built in. And we need to think about that explicitly, because I think that's important to think about whether the interests of the males and the females are aligned in the process of reinforcement. And we're now doing some sequencing of maize with different known compatibilities from different parts of the range to see, if what we've done in theory can generate a result, or it can explain what we see. So Peter, how long do I have?
Well, you have the absolute upper bound is quarter past, but it's nice to stop at, I guess, 5 past. How much longer do you want, Yaniv?
I think I'll just go through this quickly, but I'll take 5 to 10 minutes.
That'll be fine.
Great. All right. So the final result that I'm really excited about is thinking about how mechanisms of molecular mechanisms of compatibility and how plants may impact the process of how self-compatibility evolves and how gene flow evolves. And this is really cool. I don't know if I can do it. I'll see what I can do here. I won't hurry. If it's going too slow or too fast, I'll just stop. But we've recently learned this really cool system of how incompatibility works in the Solanaceae, which is the most common mechanism of self-incompatibility, which is you become self-incompatible by lacking the ability to pollinate yourself. So let's just say it's that you recognize that not that yourself and that you can't enter, but rather that you don't have the ability to. How do I explain this? My student Alex had a great analogy where he said you can think about the female part as a lock and the male part as a keychain. And you're self-incompatible when you lack the key to unlock your own lock. And that's how non-self recognition works in this collaborative this collaborative mechanism of non-self-based incompatibility. And so, but this raised a whole bunch of problems about how can you ever get a new self-incompatible type? Because if there's a new lock, nothing can unlock it. That doesn't do any good. If you get a new key, right, what good does that do you if you can't unlock anything? And through a whole bunch of clever math and helpful reviewer comments, Alex came up with this idea. You have a new key that's you have this key show up on. You have a new key. You have the ability to unlock your own key, so you're self-compatible transiently. That sucks. Your mutation selection balance at those loci, but that then allows you to have another mutation and a selective benefit to reject your own pollen. And then the whole system can go. And then we're just at this race between that key, all the haplotypes with the key, displacing everyone else, and gene conversion, moving those keys between haplotypes. And so Alex set this up as a nice model, building on the evolutionary rescue literature, and found that you can, in fact, this process can, in fact, lead to more self-compatibility, more S loci in the population. You could get this to go for quite a large number, up to 30 to 40 of them, depending on your population rate of gene conversion, which is closer to what we see in nature and exceeded what previous theory predicted. And so, there was one more thing that was so right, you can increase, but you can also decrease, because like you can have all the one key just win out if it does so before it spreads to all backgrounds. And so that was a super fun result, and we have a whole bunch of predictions that I'll talk about to anyone who cares later. But just one last thing about is that this mechanism, this non-self mechanism of self-incompatibility can actually change how you expect gene flow to happen. And so a bunch of theory, including stuff that I did before, with self-recognition said, oh, if you have a new way to a new S allele, you should adaptively introgress between species because you have this rare advantage of being able to pollinate more things. And so Alex replicated that result when you have self-recognition, but found that with non-self-recognition, you often don't see that, and lots of details matter. So this paper we just put on the bioarchive today, but that basically an invasion of a novel S allele is not guaranteed under these cases because you can suffer from pollen limitation if you're the new type and nobody has none of the native plants have the key to unlock you. And you can also don't get as strong of a benefit here because you might have a new key, but it doesn't unlock any of the natives. And so this can drive all sorts of patterns that we didn't expect before we did this theory. And most exciting, while in the self-recognition based self-incompatibility, only the one new S allele sweeps through the population, or at least increases in frequency. Here, it's kind of all or nothing, and so we expect the wholesale turnover of S alleles from one's population into another whenever one adaptively introgresses, because then you can pollinate everybody. All right, and so I wish I could put these slides up, or go read the paper. It's on bioarchive. We're super excited about it, but you can see some differences. So, for example, what were we gonna say? We'll say go read the paper. It's super fun. I don't want to spend all your time on those details. But in general, who you mate with can matter a whole lot. It can impact basic things about ecology, genetics, evolution, hybridization and its consequences, etc. And I'd like to thank the people over the years who helped me think about these ideas. And thank you all for your patience and attention. And thank Peter for the invite.
Thanks, Yaniv. You're all everybody's clapping. Do we have questions? Go ahead and unmute yourself or just type it in somewhere. Oh, Monica has a question. It is.
Yeah, so Monica says, why sulfur spread more to the higher latitude since the last glacial maximum? Is this because they have refugia in tropical areas? Is the outcrosser going to catch up later? How is the balance between outcrosser and sulfur in older, more established ecosystems? Yeah, these are all fantastic questions, and they're all things that I would like to think deeply about that sometimes go beyond our limited data collection here. So like, right, the story we're telling you, the story I would like to have you believe, but I don't necessarily believe, is that, yeah, that, all this new habitat opened up, that through serial bottlenecks, sulfurs were able to hop, skip across the landscape and make it in places where outcrossers couldn't. Now, there's a lot of other things that could happen, right? Like that is not the only thing that's different in the north in this, as you go across the globe, right? Like the Jansen-Connell hypothesis, might tell you something about, the diversity that you might expect in different places, something about, oh, maybe there's been more time for parasites to evolve and, kill these selfing species in the lower latitudes, and it hasn't yet caught up in the northern latitudes. So I think this is a good question. I can't say I know all of the answers, right? This is just kind of a vague pattern that I would like to think has something to do with how you colonize, but with any sort of broad pattern, you can think hard and come up with another explanation, and then it's our job to try to think about ways to test that and tell the difference between that hypothesis and some other. So it's a great question, and I wish I could give you a better answer.
Do you at least know, Yaniv, the last sub question is which strategy, crossing or outcrossing or selfing, is more common? Like if there's a difference in percentage of sulfurs like in the tropics versus far from the tropics?
Yeah, there's definitely, I'm quite certain, wouldn't bet, but there's a broad pattern that you have more sulfurs and also more species that reproduce asexually as you go to more northern latitudes. And that's something that you see it's pretty replicated. I think it's called geographic parthenogenesis might be the name for it. or that's the asexual version of it. And so it could be for similar reasons. But again, it's not fully worked out. It's much more of a sort of empirical pattern.
Okay, are there other questions? Also, feel free to turn on your video when you ask a question if you want.
And yeah, I recognize that was a bit of a fire hose. So yeah, Andy?
Yeah, I'll ask some questions, Yaniv. I got plenty of questions. That was really fun. Thank you for coming and speaking. So first, I'd like to ask you about the Baker's Law.
Mhm.
So what did you look at the age of those islands?
We couldn't do anything particularly sophisticated. So we did rough things like oceanic islands versus continental islands. And I forget which analysis we ended up with, but it turned out that there was nothing which showed up well enough. You know, we did like distance from mainland, and there was nothing that led us to believe that that with our data set, we could tell those different ideas apart to sort of get at the subtleties that, you know, biological details that might be underneath.
Time scale stuff. Yeah. Okay. So my other question was about reinforcement, and you kind of hinted at this. The way you were talking about the male side of reproduction. So you were talking at one point. You talked about the work with Harkness, this pollen limitation being an issue.
Mm-hmm.
From the female side, is there a cost that's analogous to polyspermy? Like, can your stigma become blocked physically with too much pollen from incompatible mates?
So
and
I have a bunch of answers to that question.
Yeah.
One is I don't know if this solves that problem because your pollen is still being clogged in this case. So if that were a problem, which it definitely is, there's some species where when you get pollinated by another species, your stigma is like, okay, I'm getting ready to do this whole fertilization thing, and so like it wastes a day with this useless pollen and then bounce back. And so in those cases, you might expect reinforcement to work in a way that might be more traditional, like you might see in animals, where maybe it's like a temporal thing or like a pollinator mediated thing where you're not getting that pollen in your way. Here, there's another magic word that you said, which is polyspermy, which is that question that I didn't address is why these incompatibilities originate at all initially. There seems to be a bunch of weird potential answers to why we see these F alleles at all. And there's some theories that it might also be, you know, a way to avoid polyspermy. There's weird results in maize where like sometimes you see that embryos and endosperm have different dads, which suggests that polyspermy might be a thing, but that's pretty speculative.
Okay, cool.
Okay, maybe we have time for one more quick question.